Hello, my name is Dr. Roderick L. Roll, and today I will be talking to you about the chromosomal basis of inheritance. The chapters that we have been covering fall into the unit called the gene concept. So I'm going to give us a brief history of some of the pioneers to this concept. Mendel's abstract proposal was created in 1860. Walter Fleming figured out mitosis in 1875. His images was subsequently published in 1882. Mr. Hertwig came in 1876. So meiosis was discovered and described by Mr. Hertwig when he observed sea urchin eggs. So this occurred in 1876. Meiosis was further described again in 1883, but now it was described on the chromosomal level. Mr. van Beneden analyzed and observed Ascaris worm eggs. In 1890, Mr. Weismann noted that 2 cellular divisions were necessary to transform one diploid cell into 4 haploid cells. This brings us to the individual that the chapter is based on, Mr. Morgan. Mr. Morgan observed a crossover event in a little small fruit fly called Drosophila. Morgan's discovery of a trait that correlated with the sex of the fly was key to the development of the chromosomal theory of inheritance. Morgan did not discover this theory, but his research led to the discovery of the chromosomal theory of inheritance. Mr. Sutton and Boveri both independently developed the chromosomal theory of inheritance in 1902. So that is a brief history of the gene concept. Mendel proposed that these genes were moving. Alleles are variations of the gene. So that's what Mendel proposed. Now, these two guys proposed that not just the gene moved, but the actual chromosome moved. So their theory states that the chromosome undergoes segregation and independent assortment. So it is not the Mendel's gene that is moving, it is the entire chromosome. So let's look at Morgan's experiment with Drosophila. Morgan noted the term wild type. The common phenotype in the fly population. If we left out a banana and the next day we captured all of the fruit fly that were associated with that banana, and we looked at the fruit flies under a stereoscope, we would see that the majority of the eyes would be red. Therefore, that had to be the wild type, the most abundant trait found in nature, wild type. Traits alternative to the wild type phenotype were called mutants. So out of the banana that was left out, if you saw maybe 5% or 2% white eyes, then that would be a mutant. So what Morgan did, he used the mutant to denote the letter to use. So because the white eye was the less prominent characteristic, he used a big W. So since he used a big W to denote the genotype, the wild type individual would be a W with a plus. In one of Morgan's experiments, he mated white-eyed males with red-eyed females. This cross is in the image above. He took a purebred red-eyed female and a purebred white-eyed male. The subsequent cross led to all offspring having red eyes. So I drew out the cross to resemble what Mendel did. By using W's, we have the wild type with the pluses and the mutant without the pluses. Every offspring in this Punnett square would be heterozygotes. Now, if we subsequently crossed those siblings, we would get a 3-to-1 ratio, 3 red eyes to 1 white eye. But what Morgan noticed after observing the sex of these flies was that 50% of the males were white-eyed and 50% of the males were red eyes. If we were to sex the flies, this is how you would distinguish which one is male versus which one is female. So like I stated, once he looked at the sex of these flies, he saw that all of the white-eyed flies were males. The standard Mendelian cross had to be modified for human genetics, for animal genetics. So this created the X-linked theory. Some genes were located on an X gene, and the X gene helped to determine sex. So this is the conclusion of Morgan's cross. You cross a red and a white-eyed purebred Drosophila and you get all red. All red are heterozygotes. Then when you do a subsequent cross of 2 F1s and get the F2, you get a 3-to-1 ratio. Morgan determined that the white-eyed mutant allele was located on the X chromosome. Notice in my image below, I have a male versus female. Notice the male only has 1 X, whereas the female has 2 X's. Morgan discovered a trait that correlated with the sex of flies. In humans and other mammals, 2 variations of sex chromosomes exist: a large X chromosome and a smaller Y chromosome. In this image, we have a replicated X chromosome and a replicated Y chromosome. The Y chromosome determines the sex in humans. A male must have at least 1 Y chromosome. A female would not have a Y chromosome. As far as homologous pairing is concerned, only a certain region of the Y is homologous with the X. In the image below, I try to draw this to illustrate it. The red long chromosome is the X, and the short black one is the Y. Only the letter E portion is going to line up and pair. The rest of it will not pair, therefore you will not get any crossing over. In this slide, we're looking at, in humans, we use the XY system. In order for you to be a female, you will have no Y chromosome. You'll have 44 autosomes and one pair of sex chromosomes, and those sex chromosomes will be XX. If you are a male, you have 44 autosomes and one pair of sex chromosomes. One will be a Y in order for you to be a male. The other can be an X. If you're going to study grasshoppers, you will look at the XO system. If you were to study chickens, you will look at the ZW system. And if you looked into bees and pollination, you will study the haploid diploid system. This slide is just further illustrating that what determines sex in humans is genes that are located on the Y chromosome. So earlier I said the Y chromosome dictates who you are. Either you're a male or a female. If you have the Y, you will be a male. So this is from the Genome Project. The sex determining region is on the Y chromosome. This is what makes you a male. The next few slides, we're going to perform some crosses. I would like for you to answer this question. What Morgan cross would yield 50% wild type and 50% mutant progeny? If you are not done, please push pause. In this cross, notice that you would have to cross a red-eyed female that is heterozygotes and a white-eyed male. If you cross those two progeny, if you cross those two individuals, you would get 50% red eye and 50% white eye. Let's perform this cross. What Morgan cross would yield 1/4 mutant progeny? If you are not complete with your assignment, please push pause. To get 1/4 mutant, you would have to cross a red-eyed male with a red-eyed heterozygote female. In this case, you would get 1 mutant, and the mutant would be male. What Morgan cross would yield all mutant progenies? If you are not complete with this assignment, please push pause. If you crossed a white-eyed female and a white-eyed male, you would get all white-eyed progeny. A gene located on either sex chromosome is referred to as sex-linked. More specifically, if it's only on the X, we would say it's X-linked. If it's only on the Y, then we would say it's Y-linked. But if it's on either one of those chromosomes, we can call it sex-linked. The smaller Y chromosome has 78 genes. And all of these genes are referred to as Y-linked genes. The larger X chromosome has 1,100 genes. These genes are referred to as X-linked genes. Many X genes are unrelated to sex, whereas most Y-linked genes are related to sex. The Y chromosome has genes that produce about 25 proteins. About half of these proteins are only expressed in the testes. So remember, you have about 10 trillion cells in your body. All cells are not going to need to express these male determining genes. Only certain cells need to produce it. So these are some of the types of cells that are in your body. So why would your brain cell need to produce these 25 male determining proteins? Why would your bones need to produce them? So majority of the production of these proteins will occur in the testicles. So the name of one of these genes is the sex determining region gene. It is responsible for the development of the testes in an embryo. Most of the genetic disorders we talked about were autosomal disorders. Autosomal dominant disorders, autosomal recessive disorders. Now we're going to talk about sex-linked disorders. Out of sex-linked disorders, you can have an X-linked or a Y-linked. So let's talk about X-linked genes. These genes will follow specific patterns of inheritance. As you can see in this image, the male only has one X, whereas the female has two X's. So in order for the female to express a disease that is linked to the X chromosome, she would have to get two of those genes. So if she only got one of those, she would be a carrier. So in the image, we have an unaffected male having sex with a carrier female. They create 4 babies. Son unaffected, daughter unaffected. 1 carrier daughter and 1 affected son. For a recessive X-linked trait to be expressed, the female needs 2 copies of the allele. And the allele is going to be a homozygous recessive, small a. But in order for the male, he only needs 1 copy. So we're going to create a new term, and it is hemizygous. So he is hemizygous, and he will express the trait. X-linked recessive disorders are many. We're only going to talk about 3: Duchenne muscular dystrophy, hemophilia, and color blindness. The first one we will look at is color blindness. X-linked recessive disorders are much more common in males because males only have to be a hemizygote to express it. So in this cross, we have a female carrier having sex with a normal male. What they produce is 50% normal, one carrier, and one colorblind individual. So remember, we have to use the terms genotypically normal versus phenotypically normal. So how many phenotypically normal individuals we produce would be 3. How many genotypically normal individuals we will produce would be 2. What cross would yield all colorblind females? Take a few minutes to try to address this question. If you're not ready to go to the next slide, please push pause. This cross would yield all colorblind female. So the male would have to have a recessive, he would have to be hemizygote. He would give all his daughters the X with the little c. The mom would have to be colorblind too. So either X she would give to her daughter would also have the little c. So colorblind female mates with a colorblind male, and you will get the right answer. All females will be colorblind. This is just a couple of different scenarios. The absence of a blood clotting factor is referred to as hemophilia. This is also a X-linked recessive disorder. Can a female with hemophilia produce a normal male? Take a few minutes to answer this question. If you are not ready to proceed, please push pause. In this cross, we will have a hemophiliac female mating with a normal male. In this case, all males will be hemophilia because the males get their Y's from dad and they get the X from mom. So mom has to have this bleeding disorder to make all males have the bleeding disorder. The 2 daughters will be carriers. Duchenne muscular dystrophy is another X-linked recessive disorder, which is plagued by progressive weakening of the muscles. The individual rarely lives past the age of 20. In mammalian females, one of the 2 X chromosomes in each cell is randomly inactivated. This occurs during embryonic development. Remember, the female has 2 X's, and if the X has 1,100 genes, she would have 2,200 genes. So one of those X's will be deactivated. So now she will have the same number of X-linked genes as we do, males. That inactivated X can be visibly seen under a microscope. It is referred to as a bar body. So the inactive X will condense to form this dark staining structure, as illustrated in the image below. Can a male have a bar body? Think about it, process it, and give me an answer. additionally to giving me an answer, write out what the individual will look like genotypically if you think the answer is yes. If you think the answer is no, then just write no. Pause the video if you're not ready to proceed. In this slide, we're looking at several different zygotes. The first one has 46 chromosomes. 44 of them are autosomes, and 2 of them are sex chromosomes. XY makes that a male. The next one is 45 with just the X. That will be a female. Neither one of those will have a bar body. If we move to the right, top right, that individual has 46 chromosomes, and the 2 sex chromosomes are both X's. One of those X's will be inactivated. It will condense and form a bar body. Under that person, that is a male. It's a male because he has a Y chromosome, but he has Klinefelter's disorder, and he has 2 X's. One of those X's will be inactivated, and it will have a bar body. Under that, to the far left, we have another female that has 3 X's. 2 of them will be inactivated, so that individual will have 2 bar bodies. So will the male that is below her. Then finally, to the far right corner, bottom corner, that female has 3 bar bodies, and so does the male. So can a male have a bar body? Yes. If a female is heterozygous for a particular gene located on the X chromosome, she will be a mosaic for that character. So this means she has big A, little a, as in reference to the sex X chromosomes. Randomly, one of those are going to be deactivated, either the big A or the little a. So to the far right, we're looking at a mosaic. Some of the cells have the big A and some of the cells have the little a. To further illustrate the randomness of this X inactivation, let's look at the tortoiseshell cat. The tortoiseshell has an allele that produces orange fur and an allele that produces black fur. This means that cat is a heterozygote. Randomly, one of them are going to be inactivated, either the orange allele or the black allele. So some parts of the cat will have black fur, some parts of the cat will have orange fur. That is how you create a tortoiseshell cat. The zygote is going to go through many mitotic divisions. And once those divisions are completed, one of those X's will be inactivated. We're not going to talk about the calico cat, but I would like for you to answer this question. Can a male tortoise shell exist? Think about everything we've talked about and then give me an answer and then explain your answer. If you can prove it by showing me the genotype or some kind of Punnett square to disprove it, please do that. If you are done, good. If not, press pause. Male tortoiseshell cats can exist, but that cat would have to be a Klinefelter's cat, and it would have to also be a heterozygote. Here is another question. Will all females have a bar body? Think about it, address it, explain it, give me the genotype, if this individual exists. If you're not complete, push pause. So the answer is no. A female that only has one X is referred to as a female that has Turner syndrome.